Hello everyone and welcome to our channel Mootechie. It's a pleasure to have you back. Today we are diving into a truly essential part of our diets, milk. We'll be exploring dairy processing technology, so get ready to learn all about what makes milk so special and how we get it from the farm to your table safely. Next, we explore our learning objectives. By the end of this lecture, you, the students, should be able to A. Understand the nutritional composition of milk and the factors affecting that composition. B. Explain the production of clean milk and the indicators of spoilage. C. Discuss the importance of milk processing. And D. Understand the major unit operations involved in the production of pasteurized and sterilized milk. These are our 4 key goals for today. Coming up, we'll start with a fundamental question: what is milk? So what is milk? It is the fluid secreted by the female of all mammalian species. Its primary function is to meet the complete nutritional requirements of the newborn of the species. Well-characterized milks that we often consume come from cows, goats, sheep, buffalo, and of course humans. Let's understand this more clearly. Milk is high in protein and essential vitamins and minerals. It's often called our most nearly perfect food. But its consumption varies widely across the globe. For instance, in Finland, the per capita consumption is about 375 liters per year. In Ethiopia, estimates suggest it's between 16 and 25 liters per year. And for context, the World Health Organization recommends about 200 liters per year. Now let's look at the nutritional content in more detail. First, let's examine the gross compositions of milk from different species. Here is a table for comparison. For human milk, total solids are 12.2%. Fat is 3.8%. Protein is 1.0%. Lactose is 7.0%. And ash is 0.2%. For cow's milk, total solids are 12.7%. Fat is 3.7%. Protein is 3.4%. Lactose is 4.8%. And ash is 0.7%. For goat's milk, total solids are 12.3%. Fat is 4.5%. Protein is 2.9%. Lactose is 4.1%. And ash is 0.8%. And for sheep's milk, total solids are 19.3%. Fat is 7.4%. Protein is 4.5%. Lactose is 4.8%. And ash is 1.0%. Following this, let's focus on the proximate compositions of cow's milk specifically. This table shows the quantitative composition of milk. For water, the limits of variation are from 85.5 to 89.5%, with a mean value of 87.5%. For total solids, it's 10.5 to 14.5%, with a mean of 13.0%. Fat varies from 2.5 to 6.0%, with a mean of 3.9%. Proteins range from 2.9 to 5.0%, with a mean of 3.4%. Lactose is from 3.6 to 5.5%, with a mean of 4.8%. And minerals range from 0.6 to 0.9%, with a mean of 0.8%. The major minerals in milk include calcium, phosphate, magnesium, sodium, and potassium. It also contains a wealth of vitamins like A, B6, B12, C, D, K, E, thiamine, niacin, biotin, riboflavin, and folates. To visualize this, we have a pie chart. It shows that milk is composed of about 87.4% water. The milk solids make up 12.6%, which is broken down into milk fat at 3.7%, lactose at 4.8%, protein at 3.4%, and other minerals, listed here as cliniads, at 0.7%. The protein is further divided into casein protein at 2.8% and whey protein at 0.6%. We also see a diagram illustrating the structure of milk at different magnifications. It shows the fat globules, the membrane surrounding them, and the serum with casein micelles dispersed throughout, giving us a glimpse into milk's complex microscopic world. Now it's important to discuss allergies related to milk. A very common one is lactose intolerance. This is the inability to digest lactose, the sugar found in milk, which causes uncomfortable side effects. So what happens? Undigested lactose is converted by the microbial flora in the intestine into lactic acid and gases. Therefore, consumption of larger quantities of milk can cause flatulence, stomach cramps, and diarrhea. So what affects what's in our milk? Let's look at the factors affecting milk composition. One major factor is breed differences. This table shows how components vary across different cow breeds. For Jersey, fat is 5.13%, lactose is 3.97%, ash is 0.76%, total solids are 14.42%. Other factors also play a big role. These include the age of the animal, the stage of lactation and pregnancy, the condition of the animal, like if it has mastitis, which is an inflammation of the udder, the method of milking, whether by hand or machine, and the completeness of milking matter. The season of the year has an influence, along with other factors like contamination and subsequent processing. This leads us to a crucial topic, the production of clean milk. This involves several key areas. First, environmental hygiene, controlling water quality, insects, and rodents. Second, the hygienic production of milk itself. This covers the areas and premises where milk is produced, ensuring animal health, following general hygienic practices including proper feeding, pest control, and managing veterinary drugs, and finally, practicing hygienic milking. Third, the handling, storage, and transport of milk. This includes using clean milking equipment, proper storage equipment and premises, and safe procedures for collection, transport, and delivery. And lastly, documentation and record keeping, which includes simple actions like weighing the milk. Let's understand this more clearly. The growth of microbes in milk is influenced by three main things: the initial cell count, the quality and kind of microbes present, and the temperature of the milk. It's fascinating to know that milk from a healthy animal already contains about 10 to the power of 3 to 10 to the power of 4 bacteria per milliliter. This is why it's so critical that milk must be chilled to 4 degrees Celsius or below as soon as it leaves the cow to slow down microbial growth. So what happens when microbes grow? Let's discuss milk spoilage indicators. The most common is souring. This is due to the transformation of lactose into lactic acid and other volatile compounds, principally by lactic acid bacteria. Then there's souring and gassiness, POT, which is often caused by the E. coli group. This indicates contamination of the milk and its products. We also have aroma production, B, which can be due to the production of flavor compounds like diacetyl. Counted on spoilage, proteolysis is another indicator. This is protein decomposition, which leads to an unpleasant odor. Ropiness is when long threads of milk are formed while pouring it. And sweet curdling occurs due to the production of a renmin-like enzyme that curdles the milk without it first souring. Now, why do we process milk at all? Well, there are 4 key reasons. 1. Preservation, to achieve a longer shelf life. 2. Reduction of health hazard, making it safer. 3. Protection, again for safety. And 4. Added value, to make it more appealing or healthier, and to improve its nutritional profile. Let's now walk through the major unit operations for the pasteurization of milk. These steps are milk receiving, preheating, separation, standardization, homogenization, pasteurization, and finally, packaging. The end goal is that carton of pasteurized milk we all know. The slide includes an image of a cheerful cartoon milk carton with a face, which helps to make the product feel familiar and friendly. The first step is the collection and reception of milk. The first thing done at reception is to determine the quantity of the milk. From weighing in, the raw milk is pumped to storage tanks to await processing. The empty churns are then conveyed to a cleaning station. The slide shows a photo of traditional milk churns being collected, which is a common sight in many parts of the world. An essential part of reception is testing the milk for quality. I. Taste and smell, checking for any deviates from the taste and smell of normal milk. 2. Cleaning checks, where the inside surfaces of farm tanks and churns are carefully inspected. 2. Sediment. This applies mainly to churns. A sample is taken from the bottom of a churn with a pipette and passed through a filter. A quality deduction is made if visible impurities are retained by the filter. Somatic cell count. This measures white blood cells. A large number, more than 500,000 cells per milliliter of milk, indicates that the cows are suffering from udder diseases like mastitis. The cell content is determined with specially designed particle counters, like a Coulter counter. Bacteria count. A simplified form of bacteria count can also be used to assess the bacteria content. Freezing point. Many dairies check this to determine if the milk has been diluted with water. Milk of normal composition has a freezing point of -0.54 to -0.59 degrees Celsius. The freezing point will rise if water is added. They also test for Y, protein content, and 7, fat content. Another simple test is measuring the density of milk. This is easy and is done with an instrument called a lactometer. A lactometer is essentially a hydrometer for milk. It can quickly and easily tell if something has been added to the milk based on changes in its density. The slide shows an image of a track lactometer, which is a specific type used for this purpose. After collection, the milk is stored in a balance tank. After the milk is collected from the farm or collection center, it's stored in refrigerated storage tanks until it's needed for processing. The balance tank is used to weigh and maintain a constant flow of liquid milk to the next operation during processing. This is crucial for a smooth and efficient production line. Now let's talk about milk separation. Newer technologies allow for both clarification and separation of milk to be done together in one operation with a three-phase separator. This means it separates out dirt, skim milk, and cream all at once. Let's understand the decreaming process itself. Separation or centrifugation is the mechanical separation of milk into cream and skim milk by using centrifugal force. This process works because it controls the efficiency of fat separation, which is possible due to the specific density differences between milk fat, which is about 0.93 grams per cc, and skim milk, which is about 1.035 grams per cc. These density differences are also related to temperature. The objectives of decreaming are to obtain a fat-reduced or fat-free milk, to concentrate milk fat for the production of high-fat products like butter, and to standardize the fat content of milk. This process is governed by Stokes' law. This brings us to the principles of centrifugal and gravitational separation. The sedimentation or flotation velocity of a particle like a fat globule can be calculated using a formula derived from Stokes' law. The formula is V_G equals frag d to the power of 2, P_P, P_T 18 eta. This calculation involves the particle diameter, the particle density, the density of the continuous phase, and the viscosity of that phase. The slide includes a diagram visualizing this sedimentation velocity and how it changes with time and particle size, showing that over time, particles of different sizes settle at different rates. So how does a centrifuge work? The centrifuge bowl has baffle inserts in the form of conical discs. This brilliant design dramatically increases the area available for sedimentation. The discs rest on each other to form a unit known as the disc stack. Radial strips called cocks are welded to the discs to keep them the correct distance apart, forming the separation channels. The thickness of these cocks actually determines the width of the channels. To visualize this, we have two figures. Figure 6.2.19 shows how in a centrifugal separator bowl the milk enters the disc stack through special distribution holes. Figure 6.2.21 shows a detailed photo of a real disc stack, where you can clearly see the distribution holes and the cocks that space the discs apart. Here's what happens inside. The cream, that is, the fat globules, has a lower density than the skim milk. Therefore, it moves inwards in the channels, towards the axis of rotation, and continues to an axial outlet. The skim milk, being heavier, moves outwards to the space outside the disc stack, and from there goes to a concentric skim milk outlet. The slide has two simple diagrams. One illustrates clarification, showing the inlet for milk and how solids are separated out. The other illustrates separation, showing how whole milk is split into two streams, skim milk moving outwards and cream moving inwards. Next, we have standardization. Standardization of fat content involves adjusting the fat content of milk by adding either cream or skim milk to obtain a specific desired fat content. There are various methods for calculating the quantities needed for this mixing. One common method is using Pearson's square. Let's work through an example. How many kilograms of cream of 40% fat must be mixed with skim milk of 0.05% fat to make a mixture containing 3% fat? The answer is obtained by drawing a rectangle and placing the fat contents on it. Subtracting the values on the diagonals gives us two numbers, C minus B equals 2.95 and A minus C equals 37. This means you would need 2.95 kilograms of 40% cream and 37 kilograms of 0.05% skim milk to obtain 39.95 kilograms of a standardized product containing 3% fat. The formulas below allow you to calculate the amounts needed for any desired quantity. Equation 1 gives you the kilograms of ingredient A and equation 2 gives you the kilograms of ingredient B. A diagram illustrates this principle of standardization. It shows a flow where 100 kilograms of raw milk is separated into skim milk and cream. These are then recombined in specific proportions, 90.1 kilograms of skim and 9.9 kilograms of cream, to create 100 kilograms of standardized milk with a precise fat content, while the leftover cream becomes a surplus product. The Pearson square is visually represented at the bottom, with the values 0.05, 40, and 3%. Another essential unit operation is homogenization. This is one of the most important steps in dairy processing. The function of homogenization is to prevent creaming, that's the rising of fat to the top of the container. It works by disrupting the fat globules. The expected result is that the milk maintains a much more uniform composition from top to bottom. homogenization often takes place at temperatures reaching 65 degrees Celsius. So how does it work? At homogenization, the milk is forced under high pressure through a very narrow gap, which splits the fat globules apart. The slide shows a diagram of a homogenizer valve. It depicts how the milk enters, is forced through the narrow gap, and exits as a homogenized product where the fat globules are now much smaller. Let's look at the principles of a homogenizer. It mechanically increases the number and reduces the size of the fat globules, taking them from an initial size of 2 to 6 micrometers down to less than 0.5 micrometers. To do this, milk is pumped under very high pressure, typically 2000 to 2500 pounds per square inch, through small mesh orifices of the homogenizer. There are single-stage and double-stage homogenizers. The process essentially emulsifies the fine fat globules, creating many new fat globule surfaces. What happens next is a bit of natural chemistry. Each newly created tiny fat globule becomes coated with a lipoprotein membrane and additional proteins from the casein in the milk. These proteins adsorb or stick to the fresh oil surface, which prevents the globules from reuniting. This is why the fat remains homogeneously distributed throughout the milk. It's also important to note that homogenization is typically done prior to subsequent pasteurization. We have a diagram that illustrates the difference between single-stage and second-stage homogenization, showing the two distinct pressure application steps that can be used for a more stable product. So what is the effect of homogenization? It has many advantages for the physical structure of milk. It creates smaller fat globules, leading to no creamline formation. It gives milk a whiter and more appetizing color. It reduces sensitivity to fat oxidation. It provides a more full-bodied flavor and better mouthfeel. And it gives better stability to cultured milk products like yogurt. However, homogenization also has certain disadvantages. Homogenized milk cannot be efficiently separated again. It becomes somewhat more sensitive to light, both sunlight and fluorescent tubes, which can result in a sunlight flavor. It can have reduced heat stability, especially with single-stage homogenization, high fat content, and other factors that contribute to fat clumping. And finally, homogenized milk is not suitable for the production of semi-hard or hard cheeses, because the coagulum will be too soft and difficult to dewater. Now, the star of the show, pasteurization. Pasteurization is a process used to kill harmful microorganisms, such as certain pathogenic bacteria, yeasts, and molds, which may be present in the milk after initial collection. This process is what significantly extends the shelf life of milk. Common time-temperature combinations are 63 degrees Celsius for 30 minutes, 72 degrees Celsius for 15 seconds. This is the most common method, known as high-temperature short time, or HTST, or 88 degrees Celsius for just 1 second. Milk's keeping quality is greatly improved, but its nutrient value is not significantly changed. Properly pasteurized milk should have a shelf life of 8 to 10 days at 5 to 7 degrees Celsius in an unopened package. Pasteurizers are typically plate-type heat exchangers. The slide shows an image of a complex industrial plate heat exchanger. It consists of many stacked corrugated metal plates forming channels for the milk and the heating or cooling fluid to pass through, allowing for very efficient heat transfer. Finally, let's touch on sterilization. This involves ultra-high temperature, or UHT, processing. The milk is heated to between 138 and 150 degrees Celsius for just 2 seconds. This process is sterile and aseptic. The great advantage is that UHT milk does not require refrigeration until the package is opened, giving it a very long shelf life. And that concludes our comprehensive overview of dairy processing technology. Thank you. See you in the next class.